Hello and welcome to another exciting lab lecture video by Dr. Armand. Hopefully everyone is staying safe, washing their hands. Today I'm wearing a nice purple shirt. Yes, I like purple, very light pastel colors. So today I decided to wear a purple shirt. To give you a little rundown of what we'll be talking about today, we'll be discussing experiment 8 lab lecture notes, synthesis of Tylenol. And then following this video, there'll be another video on how to look up some of the structures you may find on experiment 7 post lab. Because on experiment 7 post lab, you're going to be using the web CSD to search for some structures. So on that note, we'll get started. So sit back and enjoy. Oops. So last week we talked about the synthesis of aspirin. Today we're going to talk about the synthesis of acetaminophen. Again, aspirin is just to let you know, aspirin is a weak acid, and we do the esterification reaction with salicylic acid to make acetylsalicylic acid. Acetaminophen is a weak base, and you're going to see we would do a slightly different synthesis to make acetaminophen, but it's very similar to esterification reaction we did with aspirin. So just to kind of recap on some common functional groups that we use in chemistry and organic chemistry, we talked about, I think, 6 last time. We're going to introduce a few more this time. And the ones we're going to introduce are going to be ones with other heteroatoms. And so one group that's very important to note is what's called the aromatic group. And so the aromatic group consists of 6-membered rings of carbon atoms that have alternating double bonds. So if it's just one 6-membered ring with alternating double bonds, as you see here, this is called the benzene ring, which you may remember is what we used when we were drawing our saccharinate. And so the benzene ring has different forms. You can see it with the 3 double bonds shown here, or sometimes it's just drawn like this structure here. So both of these are what we refer to as benzene rings. Now when the benzene ring is attached to an R group, it's referred to as a phenyl ring. So again, benzene is when it's by itself. When it's attached to an R group, it's referred to as a phenyl group. By itself, it's benzene. Now, you can have more than one 6-membered ring with alternating double bonds. So here we have 2 6-membered rings with alternating double bonds, and this is referred to as naphthalene. Naphthalene is an aromatic group with 2 6-membered rings with alternating double bonds. Also, naphthalene is found in mothballs. If you ever go to the store and buy mothballs, you'll notice it has a very pungent odor associated with it because naphthalene sublimes at room temperature. That's why people put mothballs around their garden to keep the cats from doing their business there in the garden. So it kind of wards off the cats and other animals. Now, when you take 3 6-membered rings and fuse them together, such as we have here, so 1 is benzene, 2 is naphthalene, 3 is called anthracene. And so what I'm trying to explain here is that it doesn't have to be necessarily one ring. It can be 1, 2, 3. They even have 4 rings, etc. All of these are called aromatic rings because they have 6-membered rings with alternating double bonds. When you take organic chemistry, you'll learn more about aromatic groups, and they go a little bit more in depth. Now, last week we talked about alcohols, and we did mention in passing what a phenol is. A phenol is an alcohol, but it's when the OH group is directly attached to a benzene ring. So this OH is directly attached to a benzene ring. It's referred to as a phenol. Now again, if it's an OH on a chain or not directly attached to a benzene ring, it's called an alcohol. But when the OH is directly attached to the benzene ring, it's referred to as a phenol. And you can have more than one OH on the benzene ring. So we have aromatic, and then we have a phenol. Now let's get into some more useful functional groups. And these functional groups contain other things besides oxygen and carbon. So the first one we want to talk about is what we call an amine. This is an amine here. So an amine has a nitrogen with 3 R groups. Now these R groups can be the same or they can be different. So to give you some examples, this is called methylamine, which has 1 R group. And then you can have dimethylamine, which has 2 R groups. And again, these R groups can be the same or they can be different. Now, one thing is when they're all hydrogens, it's referred to as ammonia. That has where all the R's are hydrogen. And then we can have an amine with 3 R groups, such as this example, which is trimethylamine, 3 R groups. And so for an amine, you need a nitrogen that's attached to 1 or more R groups. Now, if it's the simplest amine, it's ammonia. So now what we're going to do is we're going to take an amine, and you can say a ketone, and we're going to make an amide. So an amide is what was shown here. It has a carbon double bond, and it has a nitrogen. And this is referred to as the amide linkage. So it's what connects the two. So an amide is a little bit different than an amine. So an amide has this C double bonded ON group. And what we're going to do, what you would have done in lab is you take an amine and convert it into an amide. One other group I want to talk about is the thiol functional group. And the thiol has the heteroatom S. And so a thiol is very similar to an alcohol, except instead of the O, you have an SH. So this SH means it's a thiol, not a alcohol. And thiols are very smelly compounds, very stinky. So these are just some additional functional groups I wanted to talk about. And remember, the whole goal of these functional groups is if you see it, can you identify what functional groups are on a compound? When you get into organic chemistry, you'll talk more about functional groups. So with Tylenol, what we're doing is we're acylating aromatic amines. So what's acylating? Acylating, we talked about this last week. We're adding this acyl group. Oops. We're adding this we're acylating, so we're adding an acyl group, which is this right here, to an aromatic amine. And the acyl group is substituted on the nitrogen atom. So remember, we're going to be starting with an amine. We're going to acylate it with something, and then we're going to acylate it on the nitrogen atom of the amide. And as we all know, it's important in over-the-counter headache remedies and also fever-reducing properties. Tylenol is a very useful drug that people use. And so in the story of Tylenol, we have to talk about 3 different amides. So in the history of Tylenol, we'll be talking about in a second, we got to talk about acetylamide, acetaminophen, and phenacetin. All of these 3 compounds play a part in the history of Tylenol. So the most common one used today is, of course, acetaminophen. So let's talk a little bit about the history of Tylenol. And depending on where you go, the histories are slightly different if you read them. And so it actually was discovered around 1886 by accident. And so the story goes that these 2 doctors, Kahn and Hepp, were doing a study to test if naphthalene can be used as a possible vermifuge. And remember naphthalene we talked about earlier? Naphthalene is an aromatic compound, and I said it's used in mothballs. Naphthalene is used in mothballs, makes it a very odorous. It's a very odorous compound. That's why they have it in mothballs. And so vermifuge means is we're seeing if it can expel worms from patients. And so early on, they found the results very discouraging. So it wasn't expelling the worms from the patients. That's not good. What do? They give up? No, they don't give up. Then Dr. Heppen reported that it had miraculous fever-reducing properties. So it wouldn't expel the worms, but it would reduce the fevers. So like any good doctor, he went to the bottle to make sure he was giving them naphthalene, and when he went to the bottle, he couldn't read the label. But he did make one interesting observation. He noticed that this sample had no odor. So the chemical in the bottle had no odor, and that's important. Why is it important? Because, as we said many times, naphthalene is a very odorous compound. It's found in mothballs. So since it didn't have this odor, he knew it wasn't naphthalene. So what did these doctors do next? What could they do? They had said it had no odor, but who are we going to call? And who do they call? They call some chemists. So chemists come to the rescue. And one of the cousins of Hepp was a chemist who helped identify the unknown, and they identified it to be acetylamide. So they weren't giving them naphthalene, they were giving them acetylamide instead, and they noticed it had fever-reducing properties. So acetylamide is actually the forerunner of what we use today, acetaminophen. We'll get to that in a second. All this came about by accident. So what happens next? Well, Kahn and Hebb published their findings in a scientific journal, and then we move on, and we find this German industrialist Karl Duisberger, who is director of research of the Bayer Company. Yes, the same Bayer Company that makes medicine today. And he had a problem. He had 50 tons of what's called for some reason I can't draw straight today, I don't know why. Paraminophenol. So paraminophenol looks something like this right here. It's what we call an aromatic amine because it has an amine group and it's attached to a benzene ring, and it's a phenol. It has a phenol group as well. And so he wanted to find a way to see or how he could make this, take this chemical and make some money for the company because it was a byproduct in another industrial process. So he read this, or he found this article by Kahn and Hepp, and he thought of a way of maybe taking para-aminophenol and converting it something similar to acetyl-anilide that had the same properties, medicinal properties. So what he decided to do is he was going to take para-aminophenol and acetylate the nitrogen atom. So he's going to add something to the nitrogen atom to make it similar to acetyl-anilide. However, back then people thought that compounds with phenols were toxic. And so since back then, when they were doing this research, since it was deemed toxic when you had a phenol on a compound, they had to come up with a way to remove the phenol from it. So what they did is they took the para-aminophenol and first they changed the phenol to an ether group. So they made it into an ether, that way it was no longer a toxic compound. And then they acetylate the amine to make it an amide. And so that's where we get what's called today, or what was called phenacetine. So phenacetine is acetyl-anilide with a ether group on it. So again, we changed the phenol to an ether because back then phenols were toxic. So phenacetine was used. It was a very highly effective fever-reducing and headache-reducing medicine. It was actually commonly found in what's called an APC tablet, which had aspirin, phenacetine, and caffeine. You could find these in little blue pouches at the checkout aisle at the grocery store. Nowadays it's called Excedrin. So if you look at the Excedrin, it has aspirin, acetaminophen, and caffeine. So we no longer use phenacetine as a medicine. And what happened is, later on they determined that not all phenol groups were toxic. So since phenol groups were not toxic, not all of them, they didn't need to ethylate the phenol group on a para-aminophenol. So today we just use what's called acetaminophen. So all we do is we acetylate the amine and leave the phenol alone, because many years later it was determined that phenols, not all of them, were toxic. So how do we make this amide? And so again, an amide is like the combination of a ketone, or as they say, a carboxylic acid, and an amine. And what we did last week is we combined a carboxylic acid and an alcohol to get an ester. That's what we did last week. And this is called, the CO is called the ester linkage, and the CN is called the amide linkage. And you may say, why can't we just take a carboxylic acid and an amine and get our amide? And the reason is when you react a carboxylic acid and an amine, you get a salt. You don't get an amide. So that's no good. So we have to come up with a different reaction scheme to take a to make an amide from an amine. And so we're going to be using something very similar to what we did last week. And so amides are formed through a reaction with what's called a primary and secondary amines. We'll talk about what those are in just a moment, with what's called an acid chloride or acid anhydrides. And this may ring a bell because last week we talked about another acid anhydride we were using, which was acetic anhydride. And so these reactions are very similar to what we call esterification reactions. So esterification, we take a carboxylic acid, we take an alcohol, it's in an acid catalyzed reaction, we form an ester. Now with amides, we take an acid chloride. An acid chloride is very similar to a carboxylic acid, but instead of an OH, it has a Cl. So you take an acid chloride, you react it with this is a primary, so we say 1 degree amine, and we get an amide, and our byproduct is HCl. That's one route. Another route is we take an acid anhydride, which looks like this. Again, we're going to react it, in this case, a primary amine. And the way we know it's primary is that it only has one R group. So it's a primary amine. And then we get an amide and a carboxylic acid. And so this is the route we would have done in lab, is we take an acid anhydride, react it with a primary or secondary amine. Again, primary and secondary mean number of R groups, so secondary would have 2 R groups. And we form an amide and a carboxylic acid. Oops, let me clear that. Now, what's the difference between primary, secondary, tertiary amides? So again, the simplest, amines. We're talking about amines here, not amides. So the simplest amine is when all the R groups are hydrogens. This is called ammonia. Now, a primary amine is when one of the R groups is replaced, one of the hydrogens is replaced with an R group. So that's a primary amine. So it only has one R group on it. Secondary amine, 2 of the hydrogens have been replaced with R groups. And then tertiary has no hydrogens on it. So it's all 3 R groups. This is very similar to alcohols. So alcohols, it depends on the number of hydrogens attached to the carbon that's attached to the oxygen. So this carbon only has one R group on it. It's called a primary alcohol. This carbon has 2 R groups, called a secondary alcohol, and this carbon has 3 R groups. So it's a tertiary alcohol. So that's how you can tell the difference between primary, secondary, and tertiary amines. In alcohols, it's the number of R groups attached to that central element. And so with amide reactions, you have to have a primary or secondary amine for the reaction to occur. So you have to have hydrogens on the nitrogen in order for a reaction to occur. So only primary and secondary amines form amides. Tertiary amines do not form amides. No reaction. You gotta have that hydrogen for it to react. So now, how would we synthesize Tylenol? So what we're going to do is we're going to take a primary amine and convert it into an amide. And the primary amine we're going to use is para-aminophenol. We react it with acetic anhydride. We get acetaminophen or amide, and we get a byproduct, acetic acid. And again, acetic acid has a very unique smell to it. So when you're doing this synthesis, you have to make sure you really wash the product in order to get all the acetic acid out of the product. And so unlike the synthesis of aspirin, and this synthesis, it likes it hot, hot, hot. That commercial, like it hot, hot, hot. Well, when you're making acetaminophen, you need to have this reaction undergoing in a hot water bath that's boiling. So it really likes it hot because you have to have it hot for this reaction to occur. And then what you would do is you would recrystallize the product and then determine the melting point and compare the melting point with the known melting point of acetaminophen. And so again, like all organic reactions, the most common calculation you do is percent yield. So here we have a student uses 2.5 grams of para-aminophenol, 3 milliliters of acetic acid. He forms 2.65 grams of the product. What's the percent yield of the reaction? And like most organic reactions, not all, but some, most of them, it's a 1-to-1 mole ratio. So again, let's dust off our limiting reactant skills. So of course we need the molar masses. We need to find the moles of each reactant. So again, acetic anhydride, you got to convert to mass, and then from mass to moles. Which one produces the fewer moles? That's our limiting reagent, and we convert that to products. So in this case, para-aminophenol produces the fewer moles of product, so it's our limiting reagent, and then we can find the theoretical yield. Once we know the theoretical yield, we can calculate the percent yield. So this is the most common calculation you do in organic chemistry. So don't forget how to do this. And so that concludes today's lab lecture over experiment 8. Shortly to follow this, you should see another video talking about the how to draw the structures you may need to look up information for on the CSD in another video after this. So on that note, I hope you enjoyed the lecture today. If you did enjoy it, make sure to click the like button. And if you learned something new, also click the like button as well. And leave a comment if you like. So thank you. Until next time.